Assalamualaikum warahmatullahi ta'ala wabarakatuh, and good afternoon everyone. So I hope all of you are doing fine and doing great. Okay, so stay safe everyone, and look at our first chapter in PSD 351, which is chromatography. Okay, for today's lecture we will cover about the paper chromatography and the thin layer chromatography, also known as TLC. Right? So first, I would like to explain about the background of the chromatography. Okay, what is chromatography? Okay, chroma from Greek words means color. Okay, while the graphy means to write. So chromatography is to write a color, which is a technique for separating mixtures into their components by differential adsorption between stationary phase and the mobile phase in order to analyze, identify, purify, and or quantify the mixture of components. So what is that means? So it means that it is an analytical technique commonly used for separating a mixture of chemical substances into its individual component, okay, so that the individual components can be analyzed. Okay, so there are many types of chromatography. We have liquid chromatography, gas chromatography, supercritical fluid chromatography. Okay, so these types of three chromatography can be divided into several. Like in GC or gas chromatography, we have gas liquid and also gas solid. So we will cover only these two types, which is gas chromatography and also the liquid chromatography. Okay. Okay, for liquid chromatography, we have ion exchange, exclusion, partition, the liquid solid absorption, paper chromatography, and thin layer chromatography. Okay, for today's lecture, we will cover these two types of chromatography. Okay, so what is chromatography? It is a technique for separating mixtures into their components in order to analyze, identify, purify, and or quantify the mixture or components. So, um, for example, okay, the application of the chromatography in our daily life, for example, in the pharmaceutical, right? So, as we all know that medicines contain many compounds, right? So, before the medicines is created, okay, they need to extract, right? Need to extract based on the sources. For example, if they want to use the organic medicines, okay, so they need to extract. So when they extract the substances, so they want to separate the compound in the substance, right? So the chromatography is one of the technique they use to analyze what is the compound in the extraction. Okay, so paper chromatography is one of the method we use to analyze simple compounds such as dyes. Okay, dye means that the ink, for example, the ink we use in our color. Okay, color, pen color, pencil color. Okay, so if we separate the color, the pigment, okay, we will see how many compound actually the dye contain, right? So we look at this diagram. For example, this diagram, it contains three mixture of compound, and then when it undergo the separation techniques, this compound will separate individually. So the level of the separation is depends on the solubility and also the polarity towards the stationary phase. Okay, how about the polarity or non-polar of the substance? Okay, and then so we can analyze, identify what type of the compound it is. We can purify and also we can quantify. Okay, we can know how many compound For example, how many compound of this compound in the mixture, how many of blue compound and how many of yellow compound, right? Okay, chromatography is used by scientists to analyze, means that we examine a mixture, its component and their relation to one another. Okay, we also identify, which is determine the identity of a mixture or components based on known components. Next, we also want to purify to isolate one of interest for further study. And the last one, we want to determine the amount of the mixture, the component present in the sample. Okay, this is the picture for the paper chromatography technique. Okay, paper chromatography is one of our lab experiment. So unfortunately, we cannot do this experiment. So this experiment actually takes quite short time, within an hour only. So what you need to do in paper chromatography, we have to set up, not the setup, the simple setup experiment only. You need to have a paper, okay, paper, kertas turas, okay, which is the standard for chromatography types. Okay, and then you need to have a dye. Okay, these types of experiment, for example, they have the red dye, black, and the green. Okay, so what you need to do is you have to mark. First, you have to draw a line, and then you put the dye on the marks here. Okay, you have to put the dye on the marks you did here. Okay, you need to mark with X. Okay, so you will have 3 X here, and then you spot the dye on the X mark. Okay, and then what you need to do is you immerse the filter paper into the water or solution. Alright, so the solution must not exceed the level of the origin line that you have made here. Okay, this is the origin line. So the water must not exceed this level. Okay, so then we can see the dye or the moving of the dye until it reach the line bottom. Okay, so you need to set whether you want to set for half an hour, 15 minutes, and so on, depend on the speed of the movement, right? Okay. In paper chromatography or in other chromatography, we have several terms like stationary phase and also mobile phase. Okay, what is actually a stationary phase? Okay, it is the substance which is fixed in place for the chromatography procedure and is the phase to which solvents and the analyte travel through or bind to. Okay, apa maksud dia? Okay, stationary phase which is it is a fixed ataupun dia adalah phase that does not move. Okay, for mobile phase, it's the analyte and solvent mixture which travel through the stationary phase. So the stationary phase actually phase that doesn't move, and the mobile phase is phase that does move. Okay, so look at the diagram back. So for paper chromatography, the stationary phase in this type of chromatography is um, things ataupun benda yang doesn't move. Okay, dia tak bergerak. So it is a paper. Okay, paper is the types of stationary phase. Okay, and the things or compound that move is mobile phase is a dye. Okay, dye ni kita consider sebagai mobile phase. Right. And the analyte is the substance to be separated during the chromatography. Okay, analyte is the substance to be separated during chromatography. Okay, some of the chromatography terms we have chromatography, which is methods of separation. A chromatograph. A chromatograph is the equipment that enable the separation. Okay, for example, we have gas chromatography. Okay, we have exclusion chromatography. Okay, so a chromatogram is the visual output of the chromatograph, which is different peaks or pattern on the chromatogram correspond to different components of the separated mixture. Okay, chromatogram means that the peak appear if you use the machine, right? Kalau macam paper, kita guna paper, dia tak ada peak, kan? If you use an instrument, you will notice the result based on the chromatogram appeared on the computer or screen. Right, so the stationary phase is the phase which is fixed in place for the chromatography procedure. Okay, it is a phase that doesn't move and it is fixed. Right, so the mobile phase is the phase which move in a definite direction. All right, this is the diagram. Okay, two more. Eluate is the mobile phase leaving the column. Okay, it is also called as effluent. So this is the eluate. Okay, so eluent is the solvent that carries the analyte, right? So we call it as the eluent. So um, this is type of the column chromatography. Okay, this is the column. So the column itself act as the stationary phase, right? So the analyte is the mixture of the compound. Okay, basically, if you use the column chromatography, you need to add a compound that you want to separate with some solvent, right? So the solvent and together with the compound you want to be separated must be dissolved. Okay, must be dissolved first, and then it called as a eluent. Okay, so when you pour the eluent into the mobile phase, okay, it turns into a mixture, kan? And then we call it as the analyte, right? So where is the mobile phase? Actually, the mobile phase is the dalam ni lah. Okay, mobile phase is here. It is the solution or also as a analyte as act as a mobile phase, right? So the component in the column will be will be move according to their affinity, right, or tendency toward the stationary phase and towards the mobile phase. Okay, the compound that is discharged first or come out first is considered as move faster and it has less tendency toward the stationary phase. All right. We look at the function of chromatography. It is the same slide as we see in the previous slide. Okay, this is the result from the paper chromatography. Okay, as we can see here, the dot, three type of dot here contains yellow, red, and green color. So when undergo the separation process, the yellow one, the yellow compound has moved faster compared to the red spot and the green spot, right? So means that the yellow constituent have a less tendency towards the stationary phase, right? So means that the yellow compound is more non-polar, right? Because of the paper chromatography, paper is a polar compound. Okay, paper is polar because it has a OH compound, SiOH compound. So paper is polar. Okay, same goes to water. Water also polar. And kalau yang non-polar contohnya minyak kan, cyclohexane tu adalah non-polar. Kalau material tu ataupun compound tu dia cepat sangat separate kan, maknanya dia tak ada kecenderungan terhadap paper ni. Okey, paper yang kita rendam ni adalah stationary phase dia adalah bersifat polar. Okey, kalau yang keluar cepat maknanya dia ni spend masa agak cepat. Okey, dia spend masa sikit dalam stationary phase. So it tends to move faster and it has less tendency towards the stationary phase. Okay, so the green one, it spend more time in stationary phase. Hence,